Okay. Good evening. I hope I'm audible. We will wait for a couple of minutes to let everybody join in, and then we will start. We'll wait for a couple of minutes. I hope the slides are visible. Yeah, thank you. Today's session is mainly about the determination of enzyme structure. This is session 2 of 12. Sorry, did you tell me anything? I couldn't catch you. Okay, welcome to the session 2 of Enzyme Sciences and Technology, weekly live interactive sessions. This is taken by Professor Vishal Trivedi of IIT Guwahati. I am Sanket from Kusuma School of Biological Sciences, IIT Delhi. So before we begin with today's discussion. So as usual, we will be discussing about whatever has been done this week on the theory, and then we will solve a couple of some questions. And as we already know that enzymes are essentially biological catalysts. And they are majorly proteins, mainly proteins. Some of them are even RNAs. So RNA enzymes are basically ribozymes. They are important in, let's say, splicing and some other activities as well. The structure of enzyme is very important to understand its function. So we have already talked about the 3D conformation and the complementarity of an enzyme with its substrate, those kind of things. Structure is very important, and today we will be mainly talking about structures of enzyme. So what enzyme does is essentially it converts substrate to product. And there is a 3D conformation of. Is there any question? 3D conformation of the substrate, and then there is a 3D conformation of the enzyme. Which has to be complementary to each other for the enzyme to function. So regarding enzyme structure, what we need to know is it's the ordered folding of polypeptide chains. Uh, so sorry, ordered folding of polypeptide chains um, to form a 3D structure. That is essentially how we see it. And essentially, there are like different degrees of structure. So first is the primary structure, which is essentially the sequence, and then there is the secondary structure, which has alpha helix and beta sheets, these kind of things. These are the secondary structures. Then these secondary structure give rise to tertiary structures, which are the arrangements of secondary structures. And then there is quaternary structure, uh, which is the arrangement of tertiary structure. And these in secondary structure, these alpha helices and beta sheets, they are also sometimes joined. So alpha helices looks something like this, and beta sheets are usually drawn like this. So they are usually joined using some loops, which are essentially unstructured loops. I would request everybody to please mute themselves. It's easy that way. Okay. So the tertiary structure essentially has the enzymatic activity. And the quaternary structure is essentially more oligomers of the protein. It might have like it might exist as a dimer or tetramer, something like that. So we will talk about it. Uh, but what we need to know is that proteins are basically polymers of amino acids. So there, we already discussed this. There are 20 amino acids, which are very important. There are some few more, which are modified amino acids, which are very rare. But 20 amino acids are usually all it takes to form a protein. And these 20, these amino acids, these polymers are joined by a covalent bond, which is called the peptide bond. Right? This is very important. If we look at the basic structure of an amino acid, it looks all the amino acids, all 20 of them, we will look at them, but all of them, they have a similar structure. They have a common structure. So there is a carbon in the middle, also known as the alpha carbon. There is a carboxyl group. So let's see. This is the carboxyl group, and there is a amine group. So this is the carboxyl group. And then there is a amine group. And then there is an R group. This is a functional group which changes in different amino acid. This R will basically change and give several different properties of an amino acid. This R is also called the side chain. We will look at it in detail when we are looking at all the amino acids. This carbon, the carbon with which R group is attached, is known as alpha carbon, or also the chiral center. All amino acids are they have this chirality. They are all alpha. I mean, they all have this chirality except glycine. In case of glycine, the R group has hydrogen. So all amino acids are chiral except glycine, which is achiral. And these, because they have chirality, they can form enantiomers. So enantiomers are basically stereoisomers, but they are not superimposable mirror images. So I think you know all of this. So based on the R group, the amino acids can be classified. So And these amino acids, they are usually represented either with a one-letter code or with a three-letter code. Both are equally used uh, and are the usual convention. First amino acid was asparagine. It was discovered in 1806 and it was isolated from asparagus. So classically, usually all amino acids have trivial names and they are usually named after the food source they were isolated from. So like asparagine from asparagus, glutamate from wheat gluten, and tyrosine from cheese. So the Greek word for cheese is tyros, so that's why the name comes as tyrosine. Glycine is known as glycine because it has a sweet taste. So this is just an example. Now, if talking about the classification, there could be a non-polar aliphatic R group. If we look at some examples here, it would be alanine, valine, leucine, isoleucine, glycine, methionine, and proline. These are non-polar aliphatic R groups. There could be aromatic R groups as well. So the examples would be phenylalanine, tyrosine, and tryptophan. There could be polar uncharged group, uncharged R group. The example would be serine, threonine, cysteine, asparagine, and glutamine. There could be a polar charged group, negatively charged group. So they have a net negative charge in the R group. So that would be aspartic acid and glutamic acid. Similarly, there could be positive charges as well, polar, obviously. It could be arginine and lysine. So these are all the amino acids. If you look at their structure, as you can see in the slide here, um, so these are the classification: non-polar side chains, uncharged and hydrophobic. So alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline are examples. Similarly, basic amino acids or negatively charged amino acids are histidine, lysine, positively charged amino acids. Histidine, lysine, and arginine are acidic amino acids would be aspartic acid and glutamic acid. Uncharged polar amino acids, which are, as we already said, serine, threonine, cysteine, tyrosine, asparagine, glutamine, and glycine. Special amino acids are modified amino acids like selenocysteine and pyrrolysine. These are present in very rare forms. They are not usually very common amino acids, but these 20 are usually the amino acids that form all the kinds of proteins that we have in our body. So after this, I think analysis of amino acid, one of the major technique is TLC, thin layer chromatography. And you can see the slide. It also depicts chromatogram of that order actually. So what we can see, different amino acids, they migrate differently because of their different R group on on the mobile phase. in this case, it would be a solvent. So in this type of a chromatographic technique, TLC, thin layer chromatography, um, it is used to separate and analyze amino acids from proteins. So you might have, maybe some of you might have done this experiment in your BSc or BTech. So here is a stationary phase, which is either silica or alumina, something like that, uh, coated on glass or aluminum foil, something like that. And then there is a mobile phase, which is usually a solvent. Ethanol could be the solvent, there could be other solvents. And this is one of the only chromatographic techniques where, there is diffusion happens from bottom to top, something like this. So if the thin layer chromatography, if this is the paper, the diffusion will happen from bottom to top. Usually in all other chromatographic techniques, it takes help of gravity, where from top to bottom we go for that kind of chromatography, but here it is different. And there is diffusion in this case. So there is a force by the mobile phase, there is a force by the stationary phase, and obviously mobile phase will allow the amino acid to move upwards, whereas the stationary phase will resist this movement. And the mobilization force in total is Fm minus Fs. That is how much force is applied on a single amino acid as of that. And there are several factors which will control this migration, right? One of them is the composition of the mobile phase. Second would be the functional group, obviously, of the amino acid. Third would be the thickness of stationary phase that will determine how much resistive force is applied. And also the functional groups on the stationary phase. So if the stationary phase, if this is a stationary phase, it has some functional groups attached to it. Could be amines. Is there any question? So that will also influence the movement of amino acids on it. So you can learn about the operations of this technique where there is spotting. Spotting is essentially in the chromatographic, the paper, you would basically make a spot of your protein. And then you run the TLC. These are the steps basically. In layer chromatogram is run, and the solvent will move towards, let's say, till here, till that time you will run this, and then you will analyze it. For analysis, there is staining required. Usually ninhydrin is used for amino acid staining. There are non-staining procedures as well. The non-staining procedures are like autoradiography. In autoradiography, the TLC plate is placed along an X-ray film for 48 to 72 hours exposure, and then that film is processed. So then fluorescence, certain heterocyclic compound give fluorescence due to the presence of conjugate double bond system. So they can be visualized in a UV chamber. So on UV excitation, they might fluoresce, something like that. So these are non-staining procedures. Talking about proteins, we already told you proteins are the polymers of amino acid. which are joined by peptide bonds. How do these peptide bonds form? So these are formed by between the carboxyl group of one amino acid with the amino group of another amino acid. So if you look at the structure again, it will look something like this: C double bond O OH. And there is some R group here. Let's make another amino acid. So between the carboxyl group of one amino acid and the amino group here, there is a decondensation reaction. Water is removed. Sorry, this is a condensation reaction. Condensation reaction or dehydration synthesis. So this is replaced by a single bond, essentially, and water is removed in the process. So this C and N bond, they cannot rotate because it has a double bond structure, because this from oxygen here through resonance, this can form a double bond, partial double bond. That's why it is not actually amenable to rotation. This CN bond is not amenable to rotation, but the CN bonds in between C alpha and C alpha N, matlab this bond and this bond, these can rotate, right? They can rotate, and the angle between them is denoted as the phi angle and the chi angles. The angle between C alpha N and C alpha C. Is all of this clear by this till this point? Anybody could raise their hand, give a thumbs up, whatever. Yeah, cool. So these angles phi and chi, they can range from minus 180 degrees to 180 degrees, essentially. And based on this, a scientist named G.N. Ramachandran, he made something known as the Ramachandran plot. And even till date, this plot is used to find out whether the conformation of the amino acid, the structure, basically the structure of a peptide, whether it is in which, whether it is basically in the allowed region, whether which form of helix is alpha helix, beta sheet, whatever is present, these kind of things can be figured out. How do a Ramachandran plot looks like? It's very simple. So this is, let's say, as I already told you, it's a plot for the chi angles and phi angles. So phi and chi are the axes, and in the middle you will have something like the zero axis. So it's 0 here and 0 here. It can range from minus 180 degrees, sorry, not Celsius. It's to plus 180 degree. Same in this axis as well. And where your amino acid lies based on the angle between C alpha N and C alpha C will actually give you certain dots on this plot. So let's say in this region, we have usually beta sheets. And let's say in this region, we have alpha helices. Specifically, let's say 210 helix, different kind of alpha helices will lie in different regions of this plot. And this is how we can verify the structure of protein. So if any amino acid residue, if it is present in between, which might be in a region like this, which could be a disallowed region, which is not possible in a system. Those kind of things can be figured out. So whenever we make models computationally, or even we go for model refinement in different kind of structural studies, we would usually refer to a Ramachandran plot. It's still very much used till date. Now, talking about the primary structure, for the primary structure of protein, that is essentially the sequence of amino acid. That is the primary structure. Now, how is the sequence of an amino acid known? First, what we need to do is, let's say this is the protein. It might have some uh, disulfide linkages as well. So first, what we do is reduce the disulfide bonds. Because disulfide bonds will interfere with the sequencing reaction. And once these disulfide bonds are reduced, it will look something like this, SH, and another will be like this, SH. This is just a rough cartoon. Please don't judge me for this. But anyway, then we will perform proteolytic cleavage with, let's say, different kind of reagents. It could be trypsin or like cyanogen bromide, something like that. We will talk about these things. And then we will perform sequencing using Edmond degradation. We will talk about it. So Edmond degradation is usually how protein sequences are determined. So there are two different proteolytic, protease. I will not say protease, but these are they basically cut proteins at different levels. Trypsin and cyanogen bromide, these are chemicals. So they will give you certain sequences. So let's say T1, T2, T3 will have some sequence, just a random sequence, let's say. So something like that, you will have different sequences. Similarly, with cyanogen bromide, you will have different uh, sequence. And what you will do is you will basically, for the actual sequence, you will see the overlaps between the two different peptides, and you will basically find the sequence. So it might be something like this based on the overlaps. So this is how structures are determined. Uh, so to break the disulfide bond, there are two approaches. Either you can go for performic acid treatment, which will cause oxidation of this disulfide bond, or there could be reduction using DTT or beta mercaptoethanol. Usually this is what is preferred. If you go for reduction and then treatment with iodoacetate, it will form carboxymethyl cysteines. So cysteines are usually the residues which have the sulfur in them. So it will form carboxymethyl cysteine, and they cannot reform the disulfide bond. So how is it done? Let's look at it. So if we have a amino acid which has a disulfide linkage in between, so to break this, there are two ways. Either performic acid you give, it will cause oxidation, and this structure will look something like this. CH, C double bond O, CH2, S Basically, this is SO3. And you will similarly get the similar structure here as well. Sorry. Another way is reduction by DTT, by using DTT, that is dihyoretol. Or beta mercaptoethanol. Is a reducing agent. They will form something like this. Let's draw it. And similar structure over there as well. And after we can take this forward and perform carboxymethylation by iodoacetate treatment. And it will cause carboxymethylated cysteine residues, which look somewhat like this. Let's draw it. So there is a carboxyl group, there's a methyl group. That's why it's called carboxymethylated cysteines. So after this, what we go for is cleavage of the polypeptide chain. So for that, we have different reagents. Let's say trypsin. We already discussed about trypsin. It's again a protein. So it cleaves after lysine and arginine. Similarly, chymotrypsin cleaves after phenylalanine, tryptophan, and tyrosine. Then there is pepsin that cleaves after leucine, phenylalanine, tryptophan, and tyrosine. And then there is cyanogen bromide, which cleaves after methionine. So these are different uh, reagents that have specific sequence where they cleave. And after that, we have sequencing, where we need to identify first the N-terminal residue. So a protein is usually ordered as N to C. This is how you want to sequence it, usually most of the time. So to identify first, we need to derive what is the terminal amino acid. So for identifying the terminal amino acid, we use something called DFNB, which is 1-fluoro-2,4-dinitrobenzene, and they will basically label the terminal amino acid, and especially at the N site, okay, in the amine group. So dinitrofluorobenzene reacts with amine group to form dinitrophenyl amino acid complex. So this is what is used. Then we go for hydrolysis of the protein, where we perform acid hydrolysis to break the protein into peptides, different peptides. And then we separate the amino acid using, let's say, HPLC or even TLC, thin layer chromatography can be used as well. And for after that, these polypeptide chains, they are sequenced using Edmond degradation. So Edmond degradation looks something like this. So phenyl isothiocyanate is used it reacts with the terminal amino acid to form phenyl carbamoyl derivative, right? As I can see, phenyl thiocarbamoyl derivative. Um, Under acidic condition that you can see in a low pH, the terminal amino acid is cleaved from the main chain as thiolinone derivative. So as you can see, this is anilino thiazolinone derivative. It is cleaved under acidic condition. This thiazolinone derivative is extracted into the organic solvent. It forms a phenylthiohydrantione amino acid, as you can see in this cartoon. Now, this PTH amino acid complex, they can be identified using HPLC, TLC. So the idea is, if this is the, let's say, sequence A, B, C, D, E, F, which these are different amino acids. So this is the nth amino acid. So what will happen? We will first go for sequencing this. So we will add this, we will form this thiazolinone derivative, take it out, sequence this. Repeat this step. This will be now in this peptide, this will be the nth amino acid. From here, we will take it out, sequence it. So based on this, we will sequence each and every um, residue, amino acid residue. And C-terminal residues, for C-terminal residues, not many methods are available, but usually from N to C orientation, we go for synthesis. I mean, not synthesis, sequencing. Then we locate the disulfide bonds as well. So we can then find out what is the exact sequence of the protein. Mass spectrometry is also used, another method for protein sequencing, very routinely used. Here, we first break the protein into different peptides, and then we go for mass spectrometry, where we analyze what is the specific amino acid present, because they will have a unique M by Z ratio, and they will have its own peak. So by that, we can also sequence it. Mass spectrometry is also used. Now, talking about secondary structure, secondary structures can be experimentally verified using two methods, either a CD spectroscopy or infrared spectroscopy. So in case of CD spectroscopy, what happens? Sorry, I'm so sorry. Yeah, CD spectroscopy. So it is also based on the fact that all the amino acids, they are chiral except one. So chirality of amino acid is the principle behind which CD works, or CD stands for circular dichroism. We will see a video where where CD is explained. So in the far UV spectra, that is from 190 to 250 nanometer range, the peptide bonds, they absorb at this range. So it's the major chromophore, and the relative orientation of peptide bonds with respect to each other, it gives us characteristic CD signal. So if we look at how CD works, let's look at the inside of the circular dichroism spectroscopy to understand how the optics is arranged. So first, there is a light source, a filter wheel, a polarizer, quarter phase plate, followed by a sample injection chamber, and the detector for detecting the light which is passing through the sample. So the light source would first give rise to light, which is then going to pass through a filter wheel, forming a monochromatic light of a single wavelength. Now that would pass through a polarizer. Now the light which is coming out of the filter wheel is unpolarized. But while it passes through the polarizer, it becomes polarized. But still it is linearly polarized. When it is passing through the lambda by 4 phase plate, it is becoming a circular polarized light. Now the sample is bombarded with the circular polarized light, and
the sample absorbs the circular polarized light in a different way, and after that, the transmitted light is detected by the detector. And from that, the circular dichroism spectra is created, which would give us important information about the secondary structure of the protein. So we know that light is an electromagnetic radiation, which has an electric field and a magnetic field. And these electric field and magnetic field vectors are mutually perpendicular to each other. And also they are perpendicular to the propagation direction. And that is how the transverse wave work, right? Now here there are many other type of polarization which are shown, like circular polarization or elliptical polarization. So what is so different about circular or elliptical polarized light? Now, in order to understand that, let's see what happens to a linearly polarized light when it passes through a quarter phase plate. So here is a linear polarized light. Now, linear polarized light occurs when the electrical field vector is oscillating in a perpendicular manner in only one plane. Okay. Now, in case of circularly polarized light, the direction of the electric field vector rotates along the propagation direction, and while it is rotating, the magnitude remains the same, but the direction is changing with the propagation time, right? So this is how a circularly polarized light is generated. But depending upon the rotation direction, either it is clockwise or counterclockwise, it is termed as left circularly polarized light or right circularly polarized light. Now, when the sample is absorbing the left and right circularly polarized light, we can get information about the concentration. So it is very similar to Beer-Lambert law. So here, the sample, which is optically active, is absorbing
Anyway, I think we don't need to know more on this. What's
the video series of Molbio explained in.
What we need to understand here is that we get results in form of ellipticity, denoted as theta. So this is basically uh, the unit would be degrees centimeter square per decamole of protein. So this is based on the characteristic wavelength. Based on the wavelength, we get theta values based on wavelength, and this is how a CD spectra would look like. And the characteristic peaks are representative of the structures like alpha helices and beta sheet. So let's say a beta sheet would probably have something, a characteristic peak like this. So CD is employed for multiple secondary structure verification, not only for proteins, even for DNA secondary structures. Let's say hairpins are formed, some other kind of structures are formed. For that also CD is used. And the abs absorbance that happen, let's say at different wavelengths like 220 nanometer range, or let's say below 210 nanometer range, they happen due to N to pi transition or pi to pi star transitions in of the electrons uh, in a peptide bond. So these are what which gives the characteristic signal, and that is how we find secondary structures in circular dichroism spectroscopy. The another form of spectroscopy is IR, infrared spectroscopy, where it again utilizes peptide bonds for the structure determination, secondary structure determination. So here there are 9 distinct absorption bands for each and every bond. So based on the stretching between carbon and nitrogen and hydrogen, they give the corresponding peaks in a IR spectroscopy. So let's say if the polypeptide conformation has alpha helix, its wave number would be in the range of 1657 to 1648. If it is a beta sheet, then it would be 1641 to 1623. If it is a unordered loop, then it would be between 1657 to 1642. And if it's a anti-parallel beta sheet, then it will have a wave number between 1695 to 1675. So if you look at an IR spectra, it's basically wave number which has a unit of centimeter inverse. And you have the spectral signals. It's usually on the negative axis, and you get peaks like this. These are characteristic IR peaks, and each peak will determine a specific, uh, signal. So it could be carbon nitrogen stretching, which will have a specific peak, or a nitrogen hydrogen bond that will have, or carbon carbon double bonds, carbon oxygen bond, something like that. After that, we'll talk about the tertiary structure. So to determine the tertiary structure, so once we know the sequence of the protein, now we know what are the secondary structures, alpha helices, beta sheets, and loops which are present in the protein. We now need to find out what is a tertiary structure. That is, how are these alpha helices, beta sheets, everything is arranged. So it can be done in two ways, actually. One is the experimental procedure, which we will discuss, and another one is non-experimental. So there are various computational approaches also. So in case of computational approach, this is non-experimental, is basically computational. We perform molecular modeling. In case of experimental approach, so you get the protein, you express the protein, purify it, then you can either form crystal, or you can keep it as the protein only in solution form. So protein expression. So if it forms a crystal, you will go for crystallography, uh, which is again very well known, but forming crystals of protein is difficult. So sometimes when it is not possible, people go for something called NMR. It's also known as it's done in the protein solution. Apart from that, without forming crystals, you can also do electron microscopy for large proteins. The best way of or to get the best resolution of a protein, X-ray crystallography is the best way to go about it. And after, with all of this, you will basically resolve the protein structure, and you will get the. So we'll talk about each and every method. So talking about crystallography, it's it's uh, it's it's a very high resolution microscopy. So it helps us visualize protein at atomic levels. If you have seen a PDB database and different protein structures that are deposited there, which are most of them are done with X-ray crystallography or NMR. So the resolution that we get with this kind of technique is around 3 to 4 angstroms, which is really good. For NMR, we get a little bit resolution on a little bit lower resolution, so that would be maybe 10 angstroms or 12 angstroms, something like that. So the higher the number, the lower the resolution. So this is for NMR, let's say. Um, Talking about microscopic techniques, we know that microscopic techniques could be light can be used, or electrons can be used, or X-ray can be used. So the wavelength for each of them, so for light, it's like 300 nanometers. Electron would be 10 nanometer. and X-ray would be 0.1 nanometer. For light microscopy is used to see the cells or the organelles in a cell, something like that. Electron microscopy is used to find out the architecture of cell, and some large proteins are can be visualized using electron microscopy. Although there is a new, very booming field of cryo-electron microscopy, which is employed for finding out the structures of proteins. Because the main problem with crystallization is making the crystal itself. It's very difficult to do that, and all proteins do not form crystal. So cryo-EM is also used. You will see certain structures coming up recently using cryo-EMs. X-ray crystallography, which has the least wavelength, you can get the atomic details of the protein. Now, if we look at the individual steps, so first you would go for protein purification. How to do that, we'll see that very soon. Then you go for crystal formation. These crystals will basically diffract to X-rays. And then we will collect this diffraction data. Then we will resolve the structure. And finally, we will make model. Resolution of structure is done using a computational program. Model building and refinement will lead to the final protein structure. How to go about overexpression of a protein? Essentially, you would clone the gene of interest. So clone the gene which leads to the protein. So you will clone this in a vector, right? And these vectors are expression vectors. A very commonly used vector for protein purification, protein overexpression in different labs is the PET series vector. You can read about it. There are different other vectors also. So these different vectors are designed keeping in mind different kind of proteins that we need to synthesize. Certain proteins are, let's say, they are cytosolic proteins, so they can be easily expressed. Certain proteins, they are encoded using rare codons. So in that case, you require those rare codons to be present in your bacteria. So these are usually bacterial expression systems. So sometimes you take the gene from humans, then express the same gene in a bacteria. So let's see how it is done. So you will clone the gene first, then you will perform transformation. Transformation is basically insertion of this DNA into a bacteria. As I told you, these are usually E. coli strains. E. coli strains. So again, bacterial systems, various types of bacterial systems are there for protein expression. Very commonly used is BL21. Then there is Rosetta strain, and there are other strains also. C41, C42, something like that. These are different E. coli strains. They have different features for specific protein to deal with specific protein problems. You can actually read about them in your free time. These are not very important with the context of the course. Then we perform induction. Now, again, one thing that I forgot to tell you about the vector, all these PET series vectors, they are usually lac operon regulated. So if you remember, if you know the concept of lac operon, there is a lac operator, lac repressor, these kind of things are present. Why is this present? Because in the system, you only want your protein or ideally in biological system, you cannot make that happen, that you only want your protein to be expressed, but you want overexpression of the majority of it should be your protein. So these lac controlled promoters, uh, So basically what happens in the bacteria, there is a lack of peron that will cause T7 RNA polymerase uh, to basically control the promoter in your vector, and you will only get your own protein. So to induce this, we can use lactose, sorry, but lactose is not usually used. What is more used is IPTG. So you would must have heard about it. IPTG is a analog of lactose, works similarly. Lactose, on the other hand, would be used up by the bacteria. This is not used up. Then you get the protein, and then you purify the protein. So protein purification is a completely different process. We are not talking about it. So talking about the methods here, how the crystal is formed, there are two ways. One is the hanging drop method, another one is the sitting drop method, where we have cover slips in both the case. And then there is a drop of protein. And below you will have the reservoir of mother liquor. And it's basically similar in either cases. But this hanging drop will essentially form a crystal. Um, Eventually. And this is done in a 24 well plate usually. These crystals are good because they are small in size. There are no defects usually in the crystal because there are no air bubbles, no cracks, nothing. Then we collect the data. Now this diffraction data is collected, um, and we get something called the electron density map. Electron density map. This electron density map contains information about where electrons are present, and based on this, we deduce the structure. Now, this phase information we can get through, there are different ways. One is molecular replacement. So what happens in molecular replacement? If you have a homologous protein which has greater than 40% sequence similarity, then you would essentially look at the structure of that protein, and you will, so let's say that protein is called the template protein and your protein is called the target protein, and you would see the structure of the template protein, and you will see what is the difference which is present in your target protein, and you will basically replace the molecules which are different and try to figure out, get the structure. So that is molecular replacement. Another way is multiple isomorphous replacement, where you essentially soak the protein in a heavy metal, which changes the diffraction pattern. Um, then there is another way, that is multi-wavelength anomalous dispersion. So there is a synchrotron radiation source which will convert methionine to selenomethionine, and it will again change the diffraction data. And based on this, when you get this diffraction data, you get the electron density map, it is then resolved to form the 3D structure of protein. Another way of going for a solution of a protein structure is NMR spectroscopy, that is nuclear magnetic resonance. So it measures the atomic distance between nuclei, basically. Um, this is performed because the protein is difficult to form crystals, let's say, which is mostly the reason. And to study the dynamics of protein, also we can, because this is carried in a solution form, the protein is responsive to, let's say, substrate inhibitors, any kind of allosteric regulators that could change the protein structure. Those kind of things can be studied using NMR only, not using X-ray crystallography, because X-ray crystallography basically you form a crystal, you get a free structure, it cannot really move. But in NMR, you can get that. So for this, you can see this video.
Methods for determining atomic structures. Nuclear magnetic resonance spectroscopy. In this method, a purified protein is mixed with special solvent and inserted into an NMR probe. The protein sample is then exposed to a strong magnetic field. This makes the nuclei of certain atoms, like hydrogen, spin like tops. When the sample is probed with radio waves, the nuclei become excited and resonate. The frequencies are measured and recorded. The surroundings of the atoms determine how high or how low the frequencies are. Using computational methods, the measurements are converted into a graph that represents the frequencies as peaks with specific locations for specific atom groups. This information is further refined and combined with additional NMR experiments to determine the 3D structure.
Yeah, that's all.
Methods for determining.
So for the steps, again, here also you will go for protein purification, as we already told you how it is done, overexpression and purification. Then you would go for NMR data collection. Then a sequential resonance assignment diffraction, data collection, collection of conformational constraints, and finally structure calculation. So if we look at protein purification, we require highly purified proteins. Are around 300 to 600 microliters of them, of the protein, and the concentration should be more than zero 0.1 millimolar. And protein is usually dissolved in a buffered solution. Now, each distinct nucleus, it produces a chemical shift by which it can be recognized. So based on this resonance, based on this magnetic resonance, there's a shift which we already saw in the video as well. So this chemical shift can happen through chemical bonds or through the empty space between the nucleus that is present. So based on this, we can find out the what is the specific bonds which are present, what is the structure essentially. This is how we get the data points. Now for data points, we need to have sequential resonance assignment. What does this mean actually? Diffraction data collection. So as I told you that there is the chemical shift. So the chemical shift are mapped by sequential walking. Uh, with multidimensional NMR spectroscopy, one can develop strategies for the assignment. If the protein sequence is known, base protein sequence is basically the primary structure. If it is known, it is easy to map the chemical shifts. So you know what are the specific residues which are present. So based on that, you can basically, there are computational programs which will give you the structure from this diffraction data points, data sets. Then we need to collect the conformational constraints. What do these mean? Conformational constraints are basically geometric informations from NMR data. So there could be some distance rates, restraint, the restraint in angles or orientation restraint. So this orientation data basically gives you information about secondary structures also. So this is very important in NMR spectroscopy. Then we go for structural calculation. Sorry. That is the final step, structure calculation. So we use this data of shifts and constraints, and then we deduce the structure. For this, a computer program is used. Now, when you get the structure, you need to have certain quality measures. So for that, there is a measure called R factor, which gives you, so in case, let's say X-ray diffraction, we talk about it. In case of X-ray diffraction, R factor will give you the progress of structure refinement. So essentially, when your computer program makes the structure, it happens in different iterations. There are different steps. Also, it's like a loop which goes on. So at each step, you get an R factor. So with more number of steps which are ongoing, the R factor will be improving. The final R factor will give you information about the quality of structure. You can find data like this on the PDB website. You can search this. PDB stands for Protein Data Bank, which has basically, it's a repository for all protein structures. You can find it out there. Then there is R free, another parameter. Um, so it's basically a cross validation between observed and expected structure. So it's another parameter. In case of NMR, there are knowledge-based methods. So that means how well a structural model conforms to expectation. So here you will see the bond length and bond angle distribution, the dihedral angles, the atomic packings, hydrogen bonds, all these kind of things. You would verify whether they are uh, in their optimal conformation. So talking about the differences between X-ray and NMR, this is performed in a solid state. NMR is performed in a solution state, although there are examples of solid state NMR as well. So since it's done in a solid state, it's static. So you get only one kind of structure, only it's a freeze conformation. This is dynamic. So if there is any particular substrate or inhibitor that you want to see, you can do that. In this case, you require crystals. Here, there is no need for crystals. It can be done in solution form. This is high resolution. This is low resolution, or I would say average resolution. In NMR, certain residues, certain amino acid residues cannot be mapped also. That is also a problem. Sometimes it happens. Here, the conformation is freezed. Here, it's dynamic. So response to substrate or inhibitors can be studied. Although it can be studied in case of X-ray crystallography as well when you have these systems, but it's a freeze system. You won't see the dynamic movements as such. Molecular modeling. Now coming to the computational approaches. In case of molecular modeling, there are two ways. So one is called the ab initio modeling. So ab initio modeling is done when the it's a non-homologous. Okay, let me write it here. Non-homologous protein structure. So let's say you found a new protein, but a homologous protein does not exist in or it has not been reported. It's a very novel protein. You do not have any homologous protein in any other where the structure has been solved. That in that case we go for ab initio modeling. Then there are new protein folds. Certain protein folds that has not been observed before. In such case, we go for ab initio molecular modeling. So what you have, how to perform it? There is a protein sequence that you have. Then you will see the protein folding. Verify it using, let's Ramachandran plot. You will check the energy parameters. So these are the parameters. What are the energy conformation on those bond angles with that conformation? What are the energy parameters? The lowest energy parameter would be the most stable. And you will also see dynamics. Sometimes in case of molecular modeling, we also go for molecular dynamics using software like GROMACS, LAM, something like that. Um, and then you get the 3D structure. Right? That is how it is done. If uh, if a homologous protein is known, then we go for homology modeling, right? So in this case, the protein structure with protein sequence with homologous structure is reported in database. So it's a multi-step process, and there are various softwares for this. It is usually much faster, even computationally, because already a structure is known. You just, again, as I told you, there is a template structure and you have your target enzyme. In case of the template structure, the protein sequence information, the folding information, and the 3D coordinates all are known. In this case, the protein sequence is known, and you deduce structure based on 3D coordinates of template. So what are the steps? First is to find query sequence. Usually the database for this is NCBI. Then you select the template. How to find the template? You perform analysis like BLAST. BLAST is basic local alignment sequence tool. So BLAST will give you templates which with most similarity. So then you will go for alignment of query. Query is your own sequence with template. Then you will go for model building. And then structure validation. For structure validation, again, Ramachandran plot. Verify 3D, another software. Then there is Save Server, something like that. These server, they provide you structure validation. For model building, one software that I remember on top of my head, it's Swiss model. You can look at it, which performs homology modeling. Now quaternary. So we are done with the primary structure, the secondary structure, and the tertiary structure. Now what we can find out is a quaternary structure. Quaternary structure is arrangements of different tertiary structures. So a protein can have its, let's say a protein that exists, you purified the protein, and it can have a native molecular weight. Native molecular weight denoted as Mn, and you do this using gel filtration chromatography. And okay, so now you also get a denatured molecular weight, and that is, let's say, MD. This you get using SDS page. SDS is a detergent which denatures your protein. And you can figure out the oligomeric state using this simple formula Mn by Md that will give you an oligomeric state of the protein. So if it is a 1, then that means the protein exists as a monomer. If it is 2, then it's a dimer. It could be trimer, or you know, uh, so on and so forth. Hexamers and decamers, the protein could be in multiple forms. So this is how you find it. Another way to do it is using, techniques, chromatographic techniques, which will give you protein, which will distinguish protein based on size. So let's say like size exclusion chromatography. In this case, what you see, something like absorbance is the y-axis, the x-axis is the elution volume, or it could be time also. So you will, after sample injection, let's say a sample is injected here, you will see different peaks like this. So these peaks, if your sample is in pure form, will give you a different oligomeric state of the protein. So in case of size exclusion, for example, the peak 1 will have the highest, I would say, molecular weight. So then there is peak 2, 3, and so on. The last dilute would have the least molecular weight. And based on this, you can form a standard curve, something like this, and you can find out which is the oligomeric state of your protein. Okay, with this, we would get into questions. Are there any questions from your end by this time? If none, we will move forward. Okay, I think we can move forward. The alpha helices and beta sheets are connected in the tertiary structure of a protein through disulfide bond, unstructured loops, hydrogen bonds, or hydrophobic interaction. What would be, anybody would like to answer? Any answers?
Unstructured loops.
You are right. You are right. The type of bond used in joining amino acid in a protein is an ionic bond, hydrogen bond, peptide bond, or van der Waals bond.
Peptide bond.
Peptide bond.
Very, very, very right. The part of the amino acid that varies among the 20 amino acid is the alpha carbon, the amine group, carboxyl group, or the side chain or R group.
Side chain.
You're right. The amino acid that contains a thiol group is proline, tyrosine, serine, or cysteine. Sorry, I couldn't get you. Uh, Anybody would like to answer? Cysteine would be the answer. Yeah.
16.
Yes. Ramachandran plot helps in determining protein primary structure, identifying amino acid side chains, defining allowed rotational angles for amino acids in protein structure, or studying the protein folding kinetics. Anybody would like to answer?
Defining allowed rotational angles for amino acid.
Absolutely correct. Absolutely correct. Match the reagent with a respective cleavage point and choose the correct option. Hydroxylamine after leucine phenylalanine trip. Okay, this is a little bit confusing. Okay, the one is hydroxylamine, and two is cyanogen bromide, three is trypsin, and four is pepsin. One is okay. A is after leucine, phenylalanine, tryptophan, tyrosine. B is after asparagine. C is after lysine and arginine, and D is after methionine. Which of these would be the right answer? Let me draw it like this. It'll be easy. Any answers?
Option D.
Option
D.
B. You're right. Option B is will be the answer. So hydroxylamine after asparagine, cyanogen bromide after methionine, trypsin after lysine and arginine, and pepsin after leucine, phenylalanine, tryptophan, and tyrosine. Phenyl isothiocyanate reacts with the terminal amino group in the Edmond degradation method to give a protein fragment, a complex amino acid, a peptide bond, or a cyclic phenyl thiocarbamyl derivative. I think I got the answer. I think it's D. That's what you said. A cyclic phenyl thiocarbamyl derivative. It's right. The role of turns in protein structure is to form alpha helices, alter the direction of polypeptide chain, serving as the primary structure of protein, or to form beta sheets. Any answers? Anybody would like to answer? It is to alter the direction of polypeptide chain. Different proteins achieve similar 3D conformation despite variations in amino acid preferences in turn regions by altering the primary structure, forming alpha helices predominantly, favoring beta sheet formation, or adjusting the length and amino acid in turn regions. Anybody would like to answer? So it's adjusting the length and amino acid in turn regions. The appearance of two overlapping bands in the amide region of the FTI spectrum of a polypeptide indicates presence of beta turn conformation alone, presence of alpha helical and beta sheet conformation, presence of random coil conformation, presence of irregular secondary structures. Not getting any answers.
Presence of alpha helical and beta sheet conformation.
Yes, you're right. The vibration that primarily contributes to amide band in the IR spectrum of the protein is due to CN stretching and NH bending vibrations. OH stretching and CO bending vibration, carbonyl stretching vibration, or SH stretching and NS bending vibration.
May CM stitching and NH bending variation.
I did not get you. Please, would you please repeat?
Answer A, CM stretching and NH bending variation.
C and NH actually they contribute, but the major one here is carbonyl stretching.
I did
Sorry. The free R factor in a protein structure determination is utilized to compute R factors using only crystallographic data, measure bond angles between the crystal structure, assess the agreement between calculated and observed data, or determines RSR and RSIM values. Anybody would like to answer this? I think we discussed this.
It's a
No, actually, R free, as we discussed in one of our slides, it's to assess the agreement between calculated and observed data. Question 13, the different types of atomic nuclei recognized in NMR spectroscopy is by their mass to charge ratio, their chemical shift and coupling constants, their electronic configuration, or their refractive index. What would be the answer? Anybody would like to answer this? No answers? The chemical shift would be the answer. The critical factor for the reliability of a homology modeling is the number of experimental validations, number of databases used for alignment, length of the protein sequence, or the degree of sequence similarity between target and template protein. What would be the answer? Any answers?
D.
Number of databases. Is that your answer? Number of databases used for alignment? Okay, nobody's answering.
Degree of sequence between target and template protein.
You're right. The construction of 3D structure in homology modeling is well guided by the sequence alignment, molecular dynamics simulation, Ramachandran plot, or the loop modeling. Any answers?
Ramachandran plot.
Ramachandran plot is to verify the 3D structure. So it's basically validation. But for construction, the first step would be sequence alignment with the target protein. So yeah, sequence alignment would be the answer to this. So this is the end of my slides. Are there any questions from your end? Is there any feedback, if not any questions, that I'm going too fast, too slow, undecipherable, anything like that? Then you can tell me. How was your first assignment then? Anybody would like to answer? Could you solve it?
Yes, sir. I will solve it.
Okay, great. I hope these are helpful. If it is, I think we can stop the recording at least at this point. Yeah.